क्या कहते जब नहीं करता नहीं। अरे हेलो हेलो। तुम्हें कहाँ है? ये जलने लगता है भाई। कहाँ जल रहा है ये? इसमें ये ब्रेक करने लगता है। हाँ चलता। अरे राहत मैडम, राहत तो दूसरे को एग्जिस्ट कैसे वो दोबारा ज्वाइन नहीं कर पाए वो।
ये मैं हमको करने
हाँ तो बता रहे थे कि ऐसे करिए, ऐसे तो कर ले रही हैं। इस पे राइट क्लिक करिए, क्लिक करने मैडम आ गए। मैम बैठो बैठो। हाँ हाँ।
हेलो।
हाँ मैं तो पूछ रहा हूँ।
हेलो। Good afternoon, ma'am.
sir. Am I audible, sir?
Yeah, am I audible?
Yes, ma'am.
हाँ सर, मैम क्वाइस जा रही है। हाँ एक सेकंड मैम। हेलो। हाँ जो
Good afternoon, sir. Am I audible, sir?
Yes, you are audible. Good afternoon.
Thank you for joining, sir. So we were waiting for more participants to join, and then we will start.
Yes, ma'am. I'm there online. Just let me know whenever it is. You are ready. So how are you, ma'am?
मेरे पास वापस में आते हो मुझे चेक करते हो
हेलो आ रही है आ रही थी ना
ये इसमें आ रहा है तो ये है मुझे रहा
नहीं बैठ जाऊंगी
ये रहा है ठीक
बंद कर दीजिए
ठीक वो भी आगे भी
So kindly move the slide. We will just once check whether it is moving or not. Yes, sir, it is. It is. Yeah. Jesus. So should we start?
Yes, ma'am. We can. Am I audible? Just to
Yes,
reconfirm again.
yes, absolutely. You're audible.
Okay.
So good afternoon, everyone. The National Pharmacovigilance Week at IMS BHU commenced with great enthusiasm, marking a significant step toward enhancing drug safety and awareness. Yesterday, we have witnessed two important sessions on incorporating ADR reporting into clinical workflow, harnessing the power of crowdsourced reporting and AI by Professor Jyotirmay Kirtania, and drug and medical drug device safety alert by Professor Hitesh Mishra, sir. More than 100 attendees joined offline and online. Today, the stage was set for another engaging series of lectures. I'm honored that we have with us Professor Pradeep Dwivedi, sir, Vice Dean Academic from AIMS Jodhpur, additional professor in Department of Pharmacology. It is my honor to introduce sir. Sir has a 14-year of teaching experience in research and teaching, more than 22 crore national and international research grant in his credit, and various travel grants, various administrative responsibilities sir is having in his shoulder. Uh, Today, sir is taking a lecture on signal reporting and management, safety signal and management. I welcome you, sir. To kindly
Thank you so
proceed.
much, ma'am. Yeah, thank you so much, ma'am, for kind introduction. So, a very good afternoon to everyone. The moment I got an information that I need to connect with the BHU, it was really heartwarming because being the alumni from the institute, I always look back to the institute and I feel that something, if I can uh, give back. So I think that I'm not a very expert at this area, but since I was invited, I readily accepted this invitation that whatever may be the topic we can take up and we can certainly discuss it. But the more important is being connected with the alma mater and yes, whatever the best I could do, I'm here to do. So anyways, in this particular pharmacovigilance week, I have been assigned this safety signals and management. Just to brief you that my slides moving. Okay. So can you see the change of slide?
Yes, sir. Yes,
Change
sir.
of
We are
slide.
able to.
Okay,
Yes,
okay,
sir.
great. So there is this IPC. Here you can see that various alerts have been sent by IPC in various months, March, May, June, July, August. So I have taken one alert to begin with to talk about that how this signals are generated and how it is being managed. So here is an example of an alert which was shared to all the healthcare professionals and the the patients. They need to know about that there is an additional alert for metronidazole, which is commonly used for amoebiasis, urogenital trichomoniasis, giardiasis, and this is FDE, fixed drug eruption, that has come to the notice of the PVPI, and they are communicating to the people, particularly the one healthcare providers, to be aware about that if you come across any reaction in terms of the FDE, please do closely monitor and please do report it to the PVPI because it is strengthened against the prevalence of occurrence of that particular event in our population. So how does this all start? Is means anybody will be questioning that what is why there is a need for the safety alerts. So we all know that medicines and vaccines have changed the way in which disease are prevented and treated, and this is an exceptional thing that has happened to this earth in last 300 years, that we have seen majority of the development in the in the medicine as well as the management of the diseases among humans. So despite their benefits, medicinal products may have some unexpected effects, so that we all know, and these effects may be unfavorable, and it may range in intensity, particularly severity that we are talking about. It could be serious, it could be the frequency issue with which it may occur in a particular population. So those things are there that need to be known in order to properly prescribe the medicine. Although the overall aim of the regulator to bring out the best medicine with all positive benefits and no harms. So despite their best effort of ensuring benefit outweighs the risk, some adverse effects are not seen in the controlled environment, and some take some time to appear. It depends upon the multiple factor. We have taken an example of the metronidazole, which was launched in 1960 in France, and from then onward, it is available across the globe. So why after 65 years are we talking about the FDE? Why shouldn't we be knowing this particular adverse effect before? So that is where the purpose of signal detection, assessment, and reporting is needed. So there are things, because our habits, our eating, our environment is dynamic, it is changing. So the way the drug body interaction environment effects are there, that can be reflected even later. So that is the whole purpose of collecting ICSR and preparing PSUR. So this periodic safety update reports as well as individual case safety reports, they are being continued as long as the drug is there in the market. So the primary purpose is to ensure that the drug is not only effective but also safe as long as it is there available in market for human use. And one more important thing about the alerts is, whenever you get an alert, it means that some surveillance is happening, and that is reporting that something is happening that you should be careful about. So that's a good part of it. Then as far as the alerts are concerned, they are motivated by signals obtained from databases of pharmacovigilance system and signal warnings of ADS detected in post-marketing surveillance or the studies or the clinical trials. So the generated alert provide information to healthcare providers as well as patients about the needed regulatory actions based on the signals, such as they may restrict the use of a drug in a particular population, maybe geriatric, maybe CKD or CLD or whatever the group that may be having more frequency of the adverse effect in that particular case, or mandatory monitoring of the patient using the medicine, as the alert has been given for the metronidazole to have be careful about and reporting about the FDE. So these kind of alerts are issued in order to sensitize our HCPs and patients to be careful about that could be the possible adverse effect happening. And in worst case scenario, it may regulator may withdraw the drug from the market, considering that the risk is more than the benefit it may offer. Going back again to the pharmacovigilance, when we are talking about, it is morely a detection, assessment, understanding, and prevention of adverse effects that can happen due to any drug. So the whole saga started with the thalidomide disaster. I think that everyone must have talked about this, but my only worst purpose was to sensitize you about it. All started with the drug monitoring, and now it has grown to the whole range of the activities that are being conducted under pharmacovigilance. And the initial challenges were finding the signals of the adverse drug reactions. So for how do we collect that who is where at what point of time suffering from something that could be drug related? So that was the initial challenges, but it has changed over a period of time. And now the focus of current pharmacovigilance is to finding out the adverse effects as early as possible, and it could be a significant signal value, so that the early intervention can be done. So as far as the detection and further its assessment, evaluation and communication is concerned, so there are few things are there. Like first is hypothesis, that does this signal or does this adverse effect relate to the drug of use? If at all it is there, then how much it is, what is the strength of it based on the causality assessment, and then further its evaluation and explanation considering the various patient and environment related factor, disease related factor, it can be further taken up. So if we look at the signal management cycle, the whole thing starts with identifying a signal, identifying an adverse effect that could be possibly linked to the drug of use, and then it is validation whether it is really related or not, confirmation and analysis. Then prioritization is very important, because if any adverse effect like FD is there, FD is certainly important, but it's if it is localized cutaneous eruptions in a very uh, like not very important near to the eyes and all, not there, so that can be given lesser priority over the some fatal adverse effects that can happen because of the drug. So prioritization has to be there, that which one is more serious in nature, or the severity is more, it may cause irreversibility, or it may be prevented, may not be prevented. Based on these criteria, so you prioritize that which should be given importance for further evaluation and recommendation of the action, and then tracking of the follow-up activities are there, and then communication that we have seen as a drug alert in order to risk minimization, and this cycle goes on. So we will take up individual and we'll try to see that how it works in our setup. As far as the signal detection is concerned, so WHO UMC defines a signal. Safety signal refers to the information of new or known side effects that may be caused by medicine or the vaccine, typically generated from more than one single report of a suspected side effects. So it is important to note that a signal does not indicate a direct causal relationship between a side effect and a medicine. So that is a very clear that the signal is just a report that, okay, some possible relation might be happening because of that. But it is essentially only a hypothesis that together with the data and argument justifies the further need for further assessments. What could be a signal? A new event never seen before or suspected to be associated with a particular drug or biologic, a known event occurring in a patient group where it has not been documented before, maybe new in elderly, new in children, new in CKD, CLD, pregnant. So that could be a signal. A known event happening with a greater frequency and severity than expected. So previously it was happening in 5% of the population. Now it is the frequency has increased to the larger population, or the repetition of event happening because of the drug has increased, including the severity. What are the sources of signals? So from where do we find out that these adverse effects can be collected? So they are mostly reported from the clinical trial which are published in public domain. There are passive ways of spontaneous reporting of the adverse effect by end user or the HCP. They may be reporting it. Active monitoring system is there in order that people are watchful, and they will be looking after various databases to find out that what kind of adverse effects are raising with what drug. Then pharmacoepidemiology studies also hint about the possible correlation and the possibilities of signals of a particular drug. Adverse event reporting system is an important source of signal. Even the non-clinical studies, animal toxicities, they do indicate about when we do the assessment of the signal about the plausibility of the causality. So the source, another source could be the post-marketing surveillance, literature review, systematic review, and meta-analysis. So these are some of the sources from which the signals can be picked up. How the signals are generated? So they may be generated through qualitative analysis of the spontaneous reports, and quantitative analysis using data mining and statistical assessment. So there are various databases available, and these databases can be used for signal generation in the post-authorization period. So the one of the most common is spontaneous reporting system databases. Examples, the AMC may be connected with the will be connected with the VG-based UDRA vigilance AER. So FDA AERs is there, Sentinel. So these databases are there in which there are reporting of the adverse effects are there. These the advantages, they help in collection of the national and regional collection of the data, high sensitivity in detecting rare adverse effects, relatively inexpensive. Apart from that, there are other ways like prescription event monitoring databases are there. Large linked administrative databases are there, like healthcare databases where the Medicare, Medicaid. So these are the insurance company, they have for-profit databases where they keep the data of individuals with all usage of the drug and the effect being reported there. Apart from that, the electronic medical records EMR databases are there. So now Government of India is moving digital health mission. So a lot of hospitals are getting paperless, and electronic health records are being maintained and under ABHA and all. It is being planned is that the transferability of the data will be there. A physician will be access to see the patient's previous report done at any other hospital. So any digitization of the data gives an opportunity for its collection and availability for doing such kind of exploration. There are various safety signal regulations and guidelines. Particularly, one need to refer to the SIOM's Working Group 8 guideline. It's a very handy guideline, practical aspects of signal detection and pharmacovigilance. FDA also regularly issues guidance to the industry for good pharmacovigilance practices and pharmacoepidemiological assessment. Similarly, European Medicines Agency EMEA guidelines on the good pharmacovigilance practice module 9 is available, and that is related to the signal management. When we look at the assessment of the signals, there are qualitative signals and the quantitative signals. So qualitative assessment of the signal is particularly the traditional signal detection method derived from observation in patients. Spontaneous reporting typically involves a small number of the cases, around 3 to 9. A single well-documented case positive re-challenge may qualify as a valid signal. So evidence required depends upon the effects, nature, report quality, and supporting evidence, pharmacological data. And to be very comprehensive, there is a Bradford Hill criteria for causation, so that is being referred to for the strength of association, what is the frequency between the control versus the drug group, consistency, whether it is the drug adverse effect has been seen in other trial where the same drug was used. Was there any specificity like quinolones causing tendon rupture? That's a very specific, difficult to find in regular setup. Temporality is one of the aspects that we all look for, that whether any adverse effect was there within the duration period that is plausible for having that effect. Then biological gradients are there. Is there any dose response relationship is there with the drug and the adverse effect? Then plausibility, biological plausibility, that is there any mechanism of action that could explain that how the drug can produce that adverse effect. Then another terminology, coherence, is there. A little confusing between the plausibility and coherence is there. So plausibility tells there is a mechanism that causes an adverse event. Coherence tells there is an adverse event that has proposed a possible mechanism. So these are the reverse of one or two. And so these, apart from that, experiments are there. This refers to the non-clinical data that may support the clinical data. This could be the animal toxicology studies or in vitro tests. Not uncommonly in literature, this criteria has been mistaken to mean other clinical studies. Apart from that, analogy is there. Is there any analogous example of similar drugs causing similar AE? So these are some of the criteria for causation, and this is being utilized for generation of signals. Another method is quantitative generation of signals. So after 1990s, late 1990s, there was a huge chunk of the data. So it was humanly not possible to go through the individual ICSR, finding out the completeness of it, and a lot of things are there. Apart from that, a lot of adverse effects are reported in various case report, case series, and various journals, publication. So huge number of the data. So there was a need to do some quantitative signal analysis and developed for systematic signal detection in large spontaneous reporting systems such as VGVS, WHO, FDA, EMA, measured through relative risk or odds ratio in population databases or large spontaneous reporting system. A higher relative risk indicates a stronger signal. So quantitative signals require more cases than qualitative ones due to statistical consideration that is done here. Giving you an example that it's a two-way contingency table for the measure of disproportionality. Just an example of a 2 into 2 table. So number of target adverse reaction report. So here A is FDE due to metronidazole. So target drug is metronidazole. Target effect is FDE. Now A is FDE due to metronidazole. Then C is FDE due to any other drug. The number of the other adverse reaction reports from that drug, metronidazole, becomes B, and then number of other adverse reactions from other drugs. So I have given you hypothetical numbers. These are not the real number. That imagine that the 5 reports has been there of metronidazole causing fixed drug eruption, and there were 50 reports of other adverse effect in that database of metronidazole. When we look for the other drugs causing FDE, I gave here a number of 25, hypothetical number, and there are total reports roughly some 1,100 reports are there. The total number is there. And if this scenario is there and we do the risk relative or odds ratio analysis, it primarily comes around 4. So for relative risk, it is coming around 3.7. For odds ratio AC by BD, it is coming 4. So the threshold has been kept at 3. Although it has been very debated that it should be depending upon the situation, drug, contextual. It should not always be the 3 times of the frequency that you happen to get in a drug versus others. So that frequency, the threshold has been debated. However, as of today, it is 3 times. Other complex quantitative signal detection methods are there. Since 1990s, there was a lot of interest was there. So mainly involved the disproportionality analysis, which compares the relative reporting frequencies of adverse event across the group. What was the key assumption in disproportionality analysis was the higher reporting for a specific reaction. So if a drug induces a specific adverse reaction, it is reported more often for that drug compared to other drug. Second thing is similar reporting rates among drugs. So the extent of underreporting, which has been told, which has been cited, like underreporting is common. So here this assumption is that the extent of underreporting for the same reaction is assumed to be the same for different drugs also. So if it is there, it is common for majority of the drugs. And so we can not always say that the FD because of other drugs are underreported and FD because of metronidazole is overreported. Those could be a possibility, but this is the assumption that assume that like the underreporting or overreporting will be apparently same across the group. So valid reference pattern is there. The overall reporting rate of the adverse reaction is assumed to be a valid reference for comparing individual drug event combinations. So for theory of disproportionality analysis, there are two methodologies. One is classical frequentist, where the frequency has been taken as the center. So long-term frequency, assuming a repeatable experiment or sampling mechanism. Another, the methodology is Bayesian probability reflects a degree of belief, incorporating prior knowledge and updating it with the new information. So despite these different paradigms, whether it is classical methodology or Bayesian methodology, the fundamental calculations are similar across both approaches. Again, common feature of disproportionality analysis, I am giving you again this example because this is critical. So DA method contains complex safety data sets into 2 contingency table for each drug event combination. So the A becomes the reports for both drug and event of stress, event of interest. B, reports of drug with but not the event. C, reports of the event but not the drug. D, reports of neither the drug nor the event. So this is how the 2 by 2 table is made, and then the odds ratio and the proportional risk ratio has been reporting ratio is being calculated. So following this, there is a pattern analysis. When a drug and event are positively associated, counts tend to be higher in cells A and D. Drug and event reported together or neither presence will be there. If negatively associated, reports tend to fall in cell B and C. Drug appears without the event or vice versa. So there has to be association measures. So various metrics are used to calculate the strength of association from contingency table, such as reporting odds ratio, relative reporting, proportional reporting ratio. So this we have seen before, that there the reporting odds ratio AD by CB, by which giving the number it turned out to be 4, and for same this proportional reporting ratio A C plus D upon C A plus B, that was coming 3.7, and that was clear above the threshold for reporting it or for communication. So apart from that, there could be a relative reporting, RR, and information component. So various other methods are there for the calculation of these thresholds and signal generation. Another example I'm giving you, just a temporal plot depicting the proportional reporting ratio for isotretinoin and its association with the reports of depression. So here you can appreciate from 1985 to 1995, everything was between 1 to 2, and then in 2000 it crosses the proportional reporting ratio more than 3, and then it is the safety alert is generated. Now the people who are given isotretinoin should be watchful for the depression in the recipients, and that is how this calculations takes place. There are other enhanced quantitative methods referred to computer-aided statistical methodologies, and data mining algorithms are there. They rely mainly on the disproportionality analysis using 2 by 2 contingency table to assess the relationship between drugs and adverse event. What is the purpose? Purpose of enhanced method is to support the traditional pharmacovigilance approaches, but not to replace them. Analyze large data set systematically, but they are still being considered exploratory. When we look at the signal management process, so ICSR event reports are there from generated from the clinical trial. So serious adverse event is, or there is a post-marketing sources, serious and non-serious adverse events. Literature reports are there. From there, it goes to the signal detection in adverse event reporting system. Health authorities oblique monitoring center system, they just get activated, and those who are having the company databases, they do the signal detection. So traditional method is review of individual case safety report, aggregate analysis of the case report data using case counts, crude or adjusted reporting rate, etc. However, the data mining algorithm, they bank on the proportional reporting ratios, multi-item gamma, Poisson shrinkers, Bayesian confidence propagational neural network. So these are various algorithm and the tools which are available for the analysis of or the data mining of the data available from various sources. Then followed by the triage of output is done. So interpret within the context of all the relevant sources of safety data, disease knowledge, biological plausibility, alternate etiology for suspected adverse drug reaction. So other safety data to be monitored are like non-clinical or pharmacology study, non-interventional studies, published literatures, periodic safety reports, information on other drug in the same class, other relevant information. All these they help in signal evaluation. So after triaging, is there need for further investigation? If there is no need, monitor via the routine pharmacovigilance if signal is indeterminate, or close out if the signal is refuted. And if you believe that okay, it is rising and it may cross beyond the threshold, so that can impact the assessment and it should be prioritized, then signal evaluation should be done, case series analysis should be done, analysis of existing clinical trial data, literature search and review for that particular adverse event, drug event should be looked for, pharmacoepidemiological studies may be conducted, mechanistic studies may be conducted, additional clinical trial may be conducted to evaluate that there is a correlation of the drug event combination. So there are future direction in signal detection, evaluation and communication. So global pharmacovigilance is there. The field of pharmacovigilance is evolving into global endeavor. State-of-the-art methods and high quality evidence are essential for effective public health protection. As far as the regulatory demands are concerned, increasing regulatory demands for the formal benefit risk assessment as part of the risk minimization strategies. Pharmacovigilance data is also valuable for evaluating the effectiveness of the intervention designed to minimize identified risk. SIOM's 8 working group report, they goal for the signal detection, prioritization, evaluation and communication and implementation of the risk minimization efforts. Future directions are given to focus on advanced signal detection, evaluation and risk management intervention, emphasis on improving communication of the signal information. That is one of the important aspects. New direction in data mining algorithm. There are two issues, particularly the sensitivity and specificity. It is a dynamic field. Data mining in pharmacovigilance is a rapidly evolving area. Multiple data mining algorithms or tools have been developed for use with various spontaneous reporting samples. So they could be VG, Flow, WHO, FDA, EMEA, or any other databases. The problem with the tool data mining algorithms is limited work on the sensitivity, specificity, and positive predictive value of this data mining algorithms are there. So there is a definite need for a thorough evaluation of the signal threshold criteria and their impact on DMA performance. As I told you, this signal threshold has been taken as a 3, but it is often debated that it should not be 3 in all the cases. It may even increase to 5, 6, depending upon the case scenario in which it is being looked for. Just to summarize that what exactly we mean by safety signal and its management. So there is a need to detect that some adverse effects are being reported. So signal detection comprises the searching for new significant safety information across various available sources. Detection may involve a review of individual case safety reports, statistical analysis, or a combination of both. Assessment of aggregated data should also be considered. Once you detect the signal, you validate it. Should take into account consideration, previous awareness, strength of evidence, and the clinical relevance using the causality assessment and the Bradford-Hill criteria that is there. Then signal confirmation is to be there, is to ensure that okay, biologically plausible is there and temporality is there, and this is the confirm that the event that is being reported is related to the drug. So a critical step to confirm the signal and escalation of signal for further signal management activities. Then signal analysis and prioritization is done. That signal should be prioritized based on the severity, seriousness, outcome, reversibility, and potential for prevention. And depending upon its public health impact, it should be faster communicated. So that is planned. So assessment and subsequent recommendation for the action. This particular part is for the regulatory assessment of all collection of the signals generated in the trials for any regulatory actions. So report should include a proposed recommendation and should be a communication to exchange all aspects of safety regarding particular signal. So this, in summary, you need to detect it, you need to validate it, you need to confirm, you need to prioritize, and you need to communicate. Based on it, take the recommendation that you can be monitoring it in a particular population, or you may be withdrawing the drug from the market. All these are the recommendation can be taken based on the conclusion drawn from that particular drug event combination. So everything starts with the treatment, and when you treat, you do realize that some side effects have happened, you report to it, then there is a collation of that information, analysis, study, and then you act based on the priority, and then you communicate to the HCP and the patient likewise in order to get the best of the drugs, making it safer its use. So thank you. I will be happy to answer.
Thank you so
I
much,
know
sir.
that it was really very fast, but
No,
yes.
sir. The comprehensive talk, sir, some examples, statistical analysis you have given, it is really insightful and will be definitely helpful for the students. Now the session is open for the question. Any question from the audience side?
So it is very important, like BHU is also AMC. So every student should be like engaged in ADR reporting, and they should be knowing the VG flow, the things are there. So I think it's a very important because they are the one who are going to sensitize their colleagues in the hostels and the future doctors. So I think they should also know that what happens once you report an ADR. So once you report an ADR, what does the collating agency are doing and how it is being done. So it's just the glimpse of the humongous activity that is done behind the curtain. But with a small effort, we can know it better. And
Yes, definitely. Being a budding pharmacologist, they should know that what happens after reporting. The reporting is a part of different stakeholders are there, nursing officers are there, clinicians are there, patient itself can report. But what happened after reporting, causality assessment and after report, these are the important aspect of pharmacologists should focus on. Any question?
so
Are we
I
on?
think that yes, ma'am.
Yes. So I think there is no questions from the audience side.
Yes.
Let them assess things.
No,
Definitely, sir.
I was requesting that you please share my PPT with all so that they
Sure.
can go through and they can add a built on it. It is the most basic thing that I could do in 2 days. So
No, it is absolutely comprehensively, collectively drafted PPT, sir. Thank you so much, sir, for your time. And we are looking forward to invite you at Banaras, sir, for something.
Definitely come. I told you that I always keep thinking about BHU that we should be contributing something to the institute from where we post graduated. So it will be an honor for us.
Sure, sir, sure. Thank you so much, sir, for your time and important contribution, insightful thought on the occasion of Pharmacovigilance Week. I'm very thankful to you, sir.
Thank you so much, ma'am. Thank you.